any case, this is pretty clearly a paraphyletic grade here. And because of that, it's pretty evident that the features that are shared by the bryophytes, this assemblage of three groups, were probably present in the common ancestor of all the land plants. And all three of these groups have a dominant haploid generation. So when you look at a bryophyte, the green photosynthetic thing that you're looking at is almost always the gametophyte, the haploid generation. And the sporophyte's either not even evident in the case of the liverworts. It's very, very tiny and embedded in the tissues of the gametophyte, or it's something that only is seen sporadically in the hornworts and mosses, as I'll show you. And you just saw in that one photograph. Another thing that's interesting about the bryophytes is that they don't have particularly good abilities to withstand, or I should say, to prevent desiccation from happening. So as the environment becomes dry, bryophytes readily dry out. But the really cool thing about them is that even though that's the case, they can withstand almost complete desiccation, or actually essentially complete desiccation, and still survive. So here you can see a bryophyte drying out. Now it's completely dry. And here you can see it in its hydrated form. And here you can see it here in its totally desiccated form, its completely dried out form. So they can go essentially from a mummified state to a completely photosynthetically active and happy state in an incredibly rapid period of time. And some in just a matter of a few minutes, they can make that transition. And part of the reason for that is they have heat shock proteins that stabilize their membranes, proteins. In general, these things are able to transition back and forth very opportunistically depending on the environment. And even though people tend to think of bryophytes as being kind of primitive, there are very few vascular plants that can do that, as we'll get to in a moment. But that's something that the bryophytes really can do well with regard to gametophyte generation. So some of these early adaptations to land that we see in all the land plants, but we don't see in their closest relatives. First of all, we have desiccation-resistant spores. I mentioned that the zygote in some of these close relatives in the green algae have the sporopollenin around their zygotes, but the sporopollenin is around the spores of land plants and prevents desiccation of those. The cuticle is basically just a waxy covering on the outside of a plant body, and that waxy covering prevents water loss to the atmosphere. The bryophytes don't have much of a cuticle. It's more pronounced on the sporophyte generation than on the gametophyte, which is the dominant generation. And the same goes with stomata. Stomata are basically pores on the outside of plants that allow for gas exchange in the face of the rest of it being covered by this cuticle. And we see the stomata on the sporophytes of mosses and hornworts, but not in liverworts. It apparently evolved after the liverwort clade split off. So this is something that didn't happen in the very earliest land plants, but it's evident widely after that point. But you need to have gas exchange, of course, for photosynthesis to occur. And the stomata regulate that. And we'll talk about how the pores are regulated in terms of their opening and closing a little bit later in the course. So multicellular gametangia are also a feature seen in all of the land plants. So we mentioned the gametophyte is the gamete-producing generation. And in land plants, the gametes are contained inside of a multicellular jacket of cells called a gametangium. So that's the term that's used. And unlike in the green algae, for example, the close relatives don't have a multicellular jacket of cells surrounding the gametes. So it's a protective outer covering for the gametes there, which could have been important to land occupancy as well. And then the embryo is something found in all of the land plants and not found in the algae, even the closest relatives. So the embryo is basically the early sporophyte. So we have a zygote, and then the zygote begins to develop, and it starts to, we have mitosis occurring. And the early sporophyte generation then, which is the embryo, is retained on the gametophyte and is nurtured by the gametophyte to some extent. When we get to in different groups of plants, it can be to a greater or lesser extent. But in any case, there is a retained embryo stage, and so there is some nurturing of the young sporophyte by the parent female gametophyte. And then there's the fungal association we talked about before. Remember the mycorrhizae associated with roots? Well, it turns out that the earliest land plants didn't have roots, and none of the bryophytes have roots, and even some of the vascular plants in the fossil record, that is, lack roots altogether, and some have secondarily lost roots. So these fungal associations with the land plant body, even before roots evolved, was really important for the early land plants and modern day land plants that lack roots, for example, to acquire nutrients, inorganic nutrients in water. And depending on, bryophytes can take water directly from the atmosphere. We'll show, I should mention that the bryophyte plant body, the gametophytes, can basically absorb water directly through all their tissues. Show you that again in a moment here. Rich secondary chemistry is another feature of all the land plants. And secondary chemistry basically is chemistry that's not involved in primary metabolism. So these are compounds that used to be thought of as waste products. And since plants don't have anuses to get, or abilities to dispose of waste in obvious ways, it was just thought that they accumulated all this junk in their bodies, and it didn't really have any function. But it's pretty clear now that secondary compounds are critical for plant physiology. They help to shield plants against UV light. They also protect plants from herbivores that would otherwise eat them. These secondary compounds often impart toxic properties to the foliage. So it's something that was important probably in early adaptation to land. All right, so we're going to first now, among land plants, look at a life cycle of bryophytes. We'll look at a moss life cycle. And this, again, is the alternation of generations. So we're going to be seeing gametophytes, which are haploid and multicellular, and sporophytes, which are diploid and multicellular. And here's the life cycle. And there's one little twist here. That is that in most mosses, but not in all of them, gametophytes generally tend to be unisexual. That is, they either produce sperm or they produce egg, but not both. Sometimes they do. Sometimes you can get both sperm and egg produced from the same gametophyte, depending on the moss species. But that's why we see two different haploid parts here, is because in this case we're looking at a female gametophyte, and in this case we're looking at a male gametophyte here. So here again we have the spores, which are haploid and drought resistant, and they're being dispersed out into the atmosphere. And we have germination of the spores. We have mitotic divisions giving rise to, at first, a filamentous body. And this is really just sort of an aside. It's not critical in understanding this life cycle, but you might see this term protonemata, and it just refers to the early part of the moss life cycle where it's basically filamentous. And then eventually we start to get development of the more mature gametophyte tissues, which we call initially a bud, And a lot of times mosses have these leafy-like gametophyte shoots. You can see that this looks like a leafy shoot. And if we take a close-up of the tips here, you can see the gametangia. Remember, multicellular gametangia, which there are two terms for these gametangia depending on whether they produce eggs or they produce sperm. And in land plants, gametangia do not produce both legs. An individual gametangium will produce either sperm or eggs, but not both. So the female gametangium that produces an egg is called an archegonium. Singularly archegonia. I'm sorry, plural is archegonia, and singular is archegonium. And then the sperm-producing gametangium is called an antheridium, or plural is antheridia. And so this antheridium is basically a sac, single cell layer in thickness, and it eventually breaks open and releases these biflagellated sperm that have two flagellae up at the front end, and they have to swim through a layer of water to get to the archegonium, which is a flask-shaped gametangium that's open at the tip. It has a neck, and it has a flask-shaped base where the egg is contained. And the sperm has to swim down that neck canal to get to the egg to fertilize it, and then we have the zygote at that point. And you can see the zygote here inside that flask-shaped base of the archegonium. It swam down through the neck canal there. And then we see the embryo. Mitoses are happening here, and we have the embryo developing inside that flask-shaped archegonium and being nurtured by the female gametophyte. And then eventually the sporophyte will continue to develop, and it's in brown here. You can see it's embedded a foot down inside of the female gametophyte from which it derives nutrients. And then there's what's called a seta, the stalk that's developing, and eventually the tip matures into what's called a capsule. And inside that capsule is the sporangium, multicellular jacket around cells that will undergo meiosis to become spores. And so here we can see a capsule that's releasing spores from the sporangium contained inside of it. There's often a little cap on the tip that falls off at maturity. And if we look at that region right there, here it is enlarged, and you can see these structures that are sticking out like teeth. This whole structure taken together is called the peristome, and it responds to atmospheric humidity. So when the atmospheric humidity is high, these structures fold back down over the tip of the sporangium and prevent the spores from coming out. And when atmospheric humidity is low, they expand out and let the spores out. So the spores are released during periods of low atmospheric humidity, which is ideal for air dispersal of the spores. So that's an innovation of the mosses in particular among the bryophytes. The peristome is only seen in the mosses. It's not seen in the liverworts or the hornworts, those other two lineages of bryophytes. But the other bryophytes have a similar life cycle in the sense that they have a dominant gametophyte generation and a relatively short-lived and completely dependent sporophyte generation that's parasitic on the gametophyte. Okay, so are there questions about this life cycle before we go on? Yeah. They're unisexual. This is a sexual cycle. There's no asexuality shown here. Mosses can reproduce asexually, but this is the sexual life cycle shown here. So this is just the male gametophyte and the female gametophyte parts of that. And sometimes there's just a bisexual gametophyte that produces both archegonia and antheridia. But it's this one that's shown a little more elaborate and more typical. All right, so let's look at some different bryophyte groups. Okay, first the mosses again. They typically have these leafy-like shoots. The green tissue here is all haploid. And here you can see the seta in a capsule of the sporophyte generation, which you see is not green. This is a parasite basically on the gametophyte generation. And here you can see the gametophyte, which is extensive and can be really long-lived. And again, these reddish sporophytes here that are that can be photosynthetic, but to a very limited extent. In fact, the stomata and cuticle the stomata are only seen on the sporophytes here, those pores that allow for some gas exchange, and there can be limited photosynthesis there. There are about 15,000 species of mosses, so it's a pretty diverse group, and it's probably really underrepresented by the current taxonomy because there's a lot of diversity that's morphologically very difficult to distinguish, and biologists are constantly finding new species of mosses using molecular data. So that 15,000 number is probably really low. There are about 9,000 species of liverworts, so it's also a really diverse group. The liverworts often have these, as you've seen, these prostrate sort of phalloid, we call them phalloid bodies that look like they're often a lobe structure that somewhat resembles those lichens that we saw earlier, but these are definitely not lichens, these are land plants. And you saw one like this in lab, I believe. They're typically with sporophytes that are embedded in the tissues of the gametophyte, but they sometimes can produce an extension of the gametophyte body that elevates part of it up above the ground level, and then the sporophytes are embedded within those structures. But the sporophytes are oftentimes reduced just basically to a sporangium, a sac of cells. And the hornworts are the least diverse. There are only about 100 species known. And they have, again, these prostrate green gametophytes, like you can see here. And they're often really slimy in texture. Nobody really likes the feeling of a hornwort. They're pretty, they have a pretty awful feel to them. They're pretty slimy organisms. But they, interestingly, have an association with a blue-green bacterium that allows them to fix nitrogen. So they're important in soil formation. And they also have a sporophyte that has indefinite growth. So unlike the sporophyte here, it's terminated by a capsule and has growth terminating at that point. The sporophytes of hornworts can grow indefinitely. And they have stomata as well. You can see some greenish cast of these. They're photosynthetic. And some phylogenetic analyses put these as sister to the land plants, and that indefinite growth of the sporophyte is something shared with, with the vascular plants, sister to the vascular plants in some analyses. All right, so economic uses of mosses are relatively limited, but one really important use is peat, which is basically deposits that are left by peat mosses, which are the genus Sphagnum, and which are found in northern and cool climates. And Sphagnum mosses have phenolic chemistry that inhibits other organisms like bacteria and fungi. They also produce conditions that are highly acidic where they grow, and also oxygen tends to be low in these habitats as well. So sphagnum bogs, as they're called, often have overall, well, they're not friendly to microbial life or fungal life, and the carbon that accumulates in those environments basically doesn't decay very quickly, and so we have massive accumulation of carbon deposits. And some of these peat bogs are actually rich sources of carbon that's used, unfortunately, as fuel in places like the UK. So there's wide harvesting of peat for use as fuel, but it also has really good use in terms of its ability to soften water or to condition soils. So it's commonly used in the nursery trade. And it has a lot of water holding capacity, and it also has a lot of antimicrobial properties. We'll get to those in a moment with regard to economic uses. But one of the most interesting things, well, as we have the drying out now of a lot of the northern environment, it's important to mention that some of the carbon that's been trapped in these moist environments as they dry out is being released into the atmosphere. And there's a real concern about a lot of this carbon ending up in the atmosphere because it's been accumulating for thousands of years and represents a pretty big store of carbon. One of the more interesting things about these bogs is their conditions that don't allow for decay very rapidly have also led to the preservation of a lot of remains of different organisms, and they're really rich sources of information about paleo environments going back thousands of years. So all kinds of biologists look at these environments, and even archaeologists looking at the remains of humans, like these so-called bog people in northern Europe, that over 1,000 of these people have been found that are almost, in some cases, almost perfectly preserved. And talk more about them next time when we get more into, and we'll then move off into looking at the vascular plants. But this is a really pretty interesting story with regard to this information from the bog people.